right, so this is one last review before we take our test on cell reproduction, just to make sure everybody's clear on a couple of the points that were made during this week. So for starters, take a look at the cell on the left, and the question that is being asked is this cell haploid or diploid? So the way that you're going to decide if a cell is haploid or diploid is that you're looking to see whether each chromosome has a homologous partner. In other words, another chromosome that is similar in size and shape. That's what you're looking for. So notice in this particular cell that we have basically 4 pairs of chromosomes. In other words, each of these chromosomes has what we would call a homologous partner. What that partner would have in common, or this would be called a homologous pair, in a karyotype, the bands would match, their size would match, the location of the centromere. In a drawing, you're usually looking for the size and the shape, because they might actually color them 2 different colors if they wanted to show you like that one came from the mom and one came from the dad, because what they don't have in common is their DNA sequences are not identical. They would have codes for the same traits. For example, they might both code for eye color, or they might both code for blood type, but that doesn't mean their actual DNA is the same, because one came from mom and one came from dad. So these, notice that these do all have a partner, so this would be a diploid cell. A haploid version of this cell would only have 1 copy of each chromosome. So you would see 4 chromosomes instead of 8, and all 4 would look different. So it would be very, very clear, because they would be different sizes and shapes, that they were not a matching pair. Now, the next question, what is the diploid number? 2N means the diploid number for this organism. So keep in mind, this was a misconception yesterday. The diploid number is the total number of chromosomes in a diploid cell. So the diploid number for this cell would be 8. The total number of chromosomes in the cell is 8. And the next question, what is N, or N means haploid. What is the haploid number for this organism? So the haploid number for this organism would be 4. That's how many chromosomes a gamete, a sperm or an egg, would have, and there would only be 1 copy from each of the pairs, okay? The next question, how many possible chromosome combinations are there after meiosis occurs? Well, if meiosis occurs, each new cell is going to randomly get 1 copy from each pair, 1 copy of chromosome 1, 1 copy of chromosome 2, 1 copy of 3, 1 copy of 4. And there's a thing called independent assortment. Independent assortment said that it was completely random which chromosome lines up on the left versus the right when meiosis happens. So if I had these 4 chromosomes, and let's say we call this one big A, and this one's little a, and this is big B, and this one's little b, when meiosis happens, you could give the chromosome we called big A and the chromosome we called big B. That's one possibility. Or you could give chromosome big A, but with chromosome little b, etc. So that was called independent assortment, the idea that that was random. And you can calculate how many possible combinations there are. The formula you need to memorize is 2 to the power of N. N being the haploid number. So for this cell that we were just working with, we said that N was 4. So the number of possible combinations would be 2 to the 4th, or 16 possible combinations of chromosomes in each gamete. So that's how you do those problems. All right, my second picture, so this is a karyotype. And I just realized my picture's sort of cut off here, so let me move it over so you can see it a little better. So this is a karyotype of a dog. So dogs are a little different than people. Obviously, they have a different number of chromosomes. The first question is this a male or a female? So what we're looking for here is we're just looking at the X and Y. So if you look over here, notice that this has an XY. So this would be a male. Their sex is determined just like ours with XX or XY. XX would be a female. The second question here is whether or what the diploid number of this cell is. So if I shrink this down a little bit so you can see the whole thing. So the diploid number, remember we said, is the total number of chromosomes in the cell when it's diploid. This is a diploid cell. Some people were misreading these. Just to clarify, the chromosomes here in this picture, they are X's. It's just that the 2 sides, the chromatids, they tend to stick together. So a lot of people were looking at these and thinking that these were just like 2 chromatids or something. These are X's. So in pair number 1 there, what you're actually seeing is this. the two X-shaped chromosomes. You can kind of see it on some of these. Like this one, you can kind of see that it's this with a little mini tip. But just a heads up on that. So here's the bottom line. Diploid number. So notice how there's 38 pairs of chromosomes. So if we double that, that's going to be 76. But don't forget the sex chromosomes. Notice that there's two more, the sex chromosomes. And so the diploid number would be 78. So for any karyotype, if you're asked what the diploid number is, and assuming it's that there's two copies of every chromosome in the picture, look at the pair numbers, right, and count how many of those there are. So in this case, there's 38. Double that, right, because there's two of each one. And then don't forget to add in the two sex chromosomes. And that's how you're going to get your total. A lot of times people mess up. They'll either give me that the answer is 38 because they just look at how many pairs here and forget about these. And they also forget to double that number because there's two copies of each one. All right, last but not least, a really quick walkthrough one last time of mitosis versus meiosis. So in this scenario, I'm doing it with 4 sets of chromosomes, or a diploid number of 8 chromosomes. So a reminder, prior to meiosis, you would have had G1, S, and G2, prior to mitosis or meiosis. And during S, just again, another reminder that before S, the chromosomes would have looked like this. You would have had 2 sticks, 2 little sticks, 2 of these white ones, and 2 of these black ones. So this is what it would have looked like before S. Then during S, synthesis or DNA replication, that's when each of these chromosomes would have gotten its matching chromatid. It would have made an extra copy of its DNA, and then they would look like X's. You can also refer to the worksheet key that I posted. I've got this all drawn out with a cell that has, I think, 4 chromosomes total instead of 8. Okay, so I'm not going to go through all the stages of mitosis, but the most important one, really, is going to be metaphase. I say that's the most important because that's the one where you're really going to see a difference between mitosis and meiosis. It's not more important than any other phase in the actual process. Here's the important thing, that in metaphase, these chromosomes are going to line up in a straight line down the middle, all 8 of them. So I didn't draw them very well, but here's your spindle fibers, and there are 8 chromosomes, all 8 lined up in a straight line. And then anaphase, they're going to split. At the centromere, these chromatids are going to become chromosomes on their own. So I'm going to go back to this. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. These cells, number 1, are identical to each other, and they're also identical to the original cell, which I've kind of messed up there, prior to S phase. The actual chromosome number has not changed. Now, has the amount of DNA changed? Yes. If this weighed, let's say, 10 picograms, each of these, the DNA only weighs 5 picograms because our chromosomes themselves are half the size that they were before. But again, this goes back to the fact that this is a chromosome, and this X-shaped thing is also a chromosome. This is just before S, and this is after S. So each new cell got an exact copy of the information. Now let's take a look at meiosis. So this is happening in skin cells, blood cells, bone cells, making exact copies of cells. The number of chromosomes isn't changing, it's just making copies. Now we're talking about meiosis. So I'm going to first, I'm not going to draw it, but just a reminder that at prophase 1, that's where crossing over would happen. And crossing over, a quick reminder, would be where chromosomes swap pieces of DNA with their matching homologous partner. And that's important because now in meiosis, the 2 chromatids, the 2 sides of a chromosome, are actually not identical to each other anymore, because once crossing over happens, each chromatid is a little bit unique. And that does not happen in mitosis, only in meiosis. All right, so we're going to draw a metaphase of meiosis 1. So this is metaphase 1 of meiosis 1. And here's the difference. This is like the most important difference between the 2 processes. These chromosomes are now going to line up with their homologous partner. So I have my 2 big ones, my 2 smaller ones, my 2 white ones, and my 2 kind of thick black ones, and they are lined up not in straight 8 in a row, but side by side with their homologous partner. And what's going to happen here is the partners are going to separate. So now we're going to get 1 big one, 1 little one, 1 black one, 1 white one, 1 big one, 1 little one, 1 black one, and 1 white one. So notice our chromosome number went from 8 chromosomes here to just 4 chromosomes here. These are now haploid. So the stage of meiosis that actually cuts the chromosome number in half is meiosis 1. Meiosis 1 reduces the chromosome number to its haploid number. So that's meiosis 1. So this is what they would look like at the end of meiosis 1. And technically, the way I have them drawn here, this is sort of metaphase of meiosis 2, the way I've set them up in the picture. So meiosis 2 is just like mitosis. They line up along the equator, and the X's are now going to split. So now you're going to get a big one, a little one, a white one, and a black one, big one, little one, white one, black one, and the same thing here, big one, little one, white one, black one. And none of these are identical to each other because of independent assortment and because of crossing over. Remember, it's completely random which one, these 2 chromosomes, going back to this, are not identical to each other. One came from your mom, one came from your dad. And it's random which one goes which way. So, you know, in fact, if I colored them 2 different colors and I just track them, so this has got a yellow one, this one's got a yellow one, and this one's got a yellow one. Over here, this one got a blue one, a blue one, a blue one. So notice how these are not the same. And then also, I didn't even show crossing over, which would have made these even further different from one another. The number of chromosomes did not change here in meiosis 2. These still have 4 chromosomes. Also, if this was spermatogenesis, each of these then could mature and become a sperm. But in oogenesis, the making of eggs, you would only make 1 egg. So just a reminder of why that is. In oogenesis, you would have your first division. All the cytoplasm pretty much goes to 1 cell. The other one's called a polar body. And then your second division, the same thing happens again. And this is also a polar body. And depending on what what book you look at, it may show this one dividing a second time. But the bottom line is, you get 1 gigantic egg that has all the, this is where all the mitochondria and all the cell parts and all the cytoplasm are going to come from. Because when the sperm fertilizes the egg, the only thing the sperm are going to donate are their 23 chromosomes. The egg is going to have, yes, a nucleus of 23 chromosomes also, but it's also going to have the mitochondria, the endoplasmic reticulum. All those other cell parts are going to come from the egg. So we make 1 egg and 3 what are called polar bodies. These just disintegrate. Even though they do have DNA in them, they're not viable. They don't become eggs. All right. And last little thing I'll go back to, again, independent assortment, the fact that it's random how they line up. Where does that happen? It happens in meiosis 1. I mean, if you want to get really specific, I suppose it's how they line up during metaphase 1 of meiosis 1, specifically. The fact that, the blue, I have that one with the blue and the yellow, I could do the same thing with the second one. Maybe this time the yellow one's on the left and the blue one's on the right. And then the third one, maybe the blue one's on the right again, and the fourth one, maybe now the blue one's on the left. And it's completely random every time, whether you give the blue copy or the yellow copy of each of these chromosomes. So that's independent assortment. That's where we're getting that 2 to the n calculation. So hopefully that clarifies some of the confusing points in the chapter. You also have, don't forget, there's 2 Kahoots. There's the quizzes, there's 2 Quizlet sets, there's all of the notes, of course. There's a key to the worksheet in the module. So you have a lot of different options of things that can help you to prepare. So I hope that you will use those and be prepared for our test next week.